New research says that
A new study shows.
New research suggests.
New studies suggest. A new study shows.
Now a new study finds that.
Hello, and welcome to the Health Files, the podcast that explores the complicated and often mysterious world of medical research. This is episode 7, In Search of a Cure. In this podcast, we will look at the mysterious process of how medical research is made, filtered through the media and into our minds, and the hiccups that happen along the way. Returning from holiday on September 3rd, 1928, Dr. Alexander Fleming began to sort out through the petri dishes he left in his office in London, each containing bacteria called Staphylococcus. Looking at the results of his experiment, he noticed something unusual on one dish. The entire surface of it was covered with little tiny colonies of bacterial growth, all except one area where just a blob of mold was growing. The blob itself wasn't that interesting though. It was the area around the blob. It was clear, as if Dr. Fleming thought the mold had somehow secreted something that stopped the bacteria from growing. Fleming was curious enough to run a few more experiments, and using this mold juice, he found that it was capable of killing a wide range of harmful bacteria. He then set his assistants the difficult task of isolating the pure compound from the mold juice that contained these specific bacteria-fighting properties. Anybody currently pursuing their PhD and having to be a teaching or research assistant just to make ends meet probably understands how they felt having been handed this task. The mold juice proved to be very unstable. The pharmaceutical giant Pfizer's chairman, John L. Smith, will later describe it as being, quote, as temperamental as an opera singer. The yields are low, the isolation is difficult, the extraction is murder, the purification invites disaster, and the testing is unsatisfactory, end quote. Because of all these things, Fleming's two assistants were only able to prepare small solutions of the crude material to work with. Nevertheless, they had enough to run some cool little experiments, and Dr. Fleming published his findings in the British Journal of Experimental Pathology in June 1929, citing the vast applications of his discovery. He decided that the main application would be in separating different types of bacteria in a mixed bacterial culture. Groundbreaking stuff. And it very well could have stayed there, unknown and forgotten, as just a curiosity, if not for Oxford researchers Howard Florey, Ernst Chain, and Charles Fletcher. You may not even have heard of this strange compound today. Luckily, they saw a much more important use for it. In 1939, just when World War II conditions were beginning to make research especially difficult, they decided to carry out a program of animal experiments to test out their hunch that there was more to this compound than just bacteria sorting. But again, they were faced with the same problem of production. In order to get enough mold juice to run their studies, the team needed to process up to 500 liters a week of mold. They began growing it in a strange array of culture vessels, such as baths, milk jugs, and food tins, which I assume is what we call Tupperware here in 2017. Florey, Chain, and Fletcher, and most likely a team of underpaid research assistants, carried out vital experiments showing that this mold juice had a much more widespread use. It could protect mice against infection from the deadly Streptococcus bacteria family, which can take credit for causing not only strep throat, as you might assume from the name, but also pink eye, meningitis, bacterial pneumonia, and flesh-eating bacterial infections. Seeing good results in mice, and at the time not really having any sort of review process or approval process for medical research, they immediately moved on to human trials. They found a 50-year-old woman who had advanced stage breast cancer and had not very long left to live. And they asked her to volunteer to take a dose of this new potential drug, not because it could potentially help her, but just to test whether it was toxic or not. She apparently agreed, and the drug was injected into her vein, and the researchers waited to see what would happen. Luckily, except for a slight fever, there seemed to be no serious side effects, and thus the search for more volunteers was on. The second person to try out the drug, and the first person who could actually potentially benefit from its properties, was a 43-year-old policeman called Albert Alexander. Despite being a policeman, the story of why he was in the hospital may surprise you. Apparently, he had scratched the side of his mouth while pruning roses in his garden and had developed a life-threatening infection with huge abscesses, which is an inflamed area in the body that is filled with pus, affecting his eyes, face, and lungs. As described in the official account of the clinical trial, quote, he was desperately and pathetically ill. There was all to gain and really nothing to lose for him during this clinical trial, end quote. The policeman received injections of the drug, and within days he was showing remarkable improvement. Unfortunately, due to the way that the mold juice drug was being developed and the need for constant injections, the researchers soon found their supplies dwindling, and they got creative. As the official account from one of the researchers states, quote, all of the patient's urine was collected, and each morning I took it to the Dunn laboratory on my bicycle so that the excreted drug could be extracted and be used again, end quote. Now, that is what I call recycling. Despite being injected the recycled drug, the policeman was showing a striking improvement, and after only 5 days, he was eating well, and there was a visible reduction in the abscesses on his face. But when the supplies of the drug finally did run out, despite best efforts, he regressed and died a few days later. Though a tragic story, this case was monumentally important. For the first time, it showed the remarkable effects of this new drug, proved its safety and continuous use in humans, and set it on a path to become one of the greatest inventions of all time. The drug, as you might have guessed, was penicillin, and it is still one of the most widely used antibiotics in the world today, treating everything from acne to STDs to pneumonia and sepsis. Though I am very tempted to make this episode about drug-resistant bacteria and the downfall of antibiotics, that is not the path we're choosing today. We will instead focus on the fascinating aspect of the story that it was okay to inject a compound a researcher extracted from mold juice in a petri dish into sick patients without any sort of oversight, approval, or not even a concrete plan of action. Clinical trials today, I hope this won't surprise anyone, are a little more complicated than that. Just as hard as it to imagine a world before antibiotics, the world without the gaze and funding of the World Health Organization or the National Institute of Health is a dystopian society that may keep us all awake at night. But bear with me here as we dive into how clinical trials came to be and what exactly goes on behind those closed doors and long, very, very long clinical reports. The world's first clinical trial is recorded in the Bible in the book of Daniel, believe it or not. This experiment, moderately resembling a clinical trial, was not even conducted by a doctor, but by King Nebuchadnezzar. I hope I got that right. During his rule, the king ordered his people to eat only meat and drink only wine because he believed that that's the best diet for them and would keep them in the best physical condition. But several of the young men of royal blood preferred to eat vegetables, shockingly, and they objected. The king allowed these rebels to follow a diet of legumes and water, but only for 10 days. When the king's experiment ended, the vegetarians seemed better nourished than the meat eaters, so the king permitted the legume lovers to continue their diet. This probably was one of the first times in the evolution of the human species that an open, uncontrolled human experiment guided a decision about nutrition and public health. Similarly uncontrolled experiments went on throughout the centuries up until 1747, when Dr. James Lind decided to do something a little different. He's considered the first physician to have conducted a controlled clinical trial of the modern era. While working as a surgeon on a ship, he was shocked by the high mortality rate of sailors from scurvy. Scurvy is rare in the developed world today, but early on it was a very serious concern. It caused anemia, exhaustion, swelling in some parts of the body, ulceration of the gums, loss of teeth, diarrhea, fever, and sometimes even paralysis. It was common among sailors, and many died of its symptoms. Dr. Lind decided that treatments of the time did not really have enough scientific ground to stand on, so he designed a comparative trial of the most promising cures for scurvy of the day. From his own account, Lind describes the following, quote, "On the 20th of May, 1747, I selected 12 patients in the scurvy on board the Salisbury vessel at sea. Their cases were as similar as I could have them. They all in general had putrid gums, the spots in lassitude, with weakness of the knees. They lay together in one place, being a proper apartment for the sick in the forehold, and had one diet common to them all: water gruel sweetened with sugar in the morning for breakfast, fresh mutton broth oftentimes for dinner, and at other times light puddings, boiled biscuit with sugar, etc. And for supper, barley and raisins, rice and currants, and wine. 2 of them were ordered to drink each a quart of cider a day. 2 others took 25 drops of the elixir vitriol 3 times a day. 2 others took 2 spoonfuls of vinegar 3 times a day. 2 of the worst patients were put on a course of seawater for some reason. 2 others had each 2 oranges and 1 lemon every day. And then 2 remaining patients took a medicine recommended by a local surgeon. The consequence was that the most sudden and visible good effects were perceived from the use of oranges and lemons. One of those who's taken them, at the end of 6 days, was fit for duty. The other was the best recovered of any in his condition, and was appointed to help with the rest of the sick. Next to the oranges, I thought the cider had the best effects." End quote. Although the results were clear, Lind hesitated to recommend the use of oranges and lemons because they were too expensive. It was nearly 50 years before the British Navy eventually made lemon juice a compulsory part of the sailors' diet, and this was soon replaced by lime juice because, again, it was cheaper. Now we know that scurvy is caused by a deficiency in vitamin C, which is promptly corrected by introducing fruit into the diet. Though we're not sure if Lind had any idea of why the fruits had the best effect, through the use of a controlled comparative experiment, he was able to test out different remedies in patients with similar symptoms in similar conditions, essentially laying the ground for all controlled studies to come. Quick side note, vitriol elixir, as he called it, is sulfuric acid and is super toxic and causes severe burns just from skin contact. I cannot imagine drinking any elixir or mixture containing it helped the poor guys. Oh my gosh, terrible. Anyway, they did the best they could back then, and ever since, clinical trials have been getting more and more sophisticated and adjusting not only to logical things like secrecy when it comes to assignment into groups, but also the need for things like patient data confidentiality, ethics committee review, as well as human rights protection. But before we delve into all that, let's get to the basics of what a clinical study is and how it is run. A clinical study, as you already might suspect, is a type of medical research using human volunteers called participants. It is mainly intended to add to medical knowledge. In a clinical trial or study, Participants follow a certain research plan, which differs study to study. It may be testing medical products, such as drugs or devices, or it could be comparing certain medical procedures just to see which one's better, or it may be even testing something as simple as changes to a participant's behavior, such as what they eat or drink. Clinical trials may compare something new to something already established in the market, or the comparison may be to a placebo, something that resembles the real medicine but contains no active ingredients, basically a sugar pill. They may even compare a drug or a change in behavior to no drug and no change of behavior. It really depends on what they're testing and what effects they're trying to isolate. When a new product is being studied, it is not usually known whether it will be helpful, harmful, or no different than the things already studied before. So there's always a significant amount of risk involved. Depending on a variety of factors, clinical trials are categorized into phases, 5 phases actually. Phase 0 is used for general exploratory studies with very little human exposure. These are helpful to get the general gist of the effects people might experience, kind of like how penicillin was first given to a woman just to see whether it was toxic or not, though that is a super extreme example that would totally not be okay today. Just making that clear. Phase 1 studies are a little more involved. They are usually conducted with healthy volunteers, and they emphasize safety. The goal is to find out what the drug's most frequent and serious side effects are, and what are the safe doses for further study. Phase 2 studies gather various types of data dealing with how effective the drug is, so it's tested in people who have certain diseases or conditions. Phase 3 studies gather even more information about the safety and effectiveness by studying different populations and different dosages, and by using the drug in combination with other drugs, trying to figure out what combination works best. And last but not least, Phase 4 studies. These take place only after the FDA has approved the drug for marketing. They include final data collection and some extra requirements requested by the study sponsor. Every clinical study is led by someone called the principal investigator, basically the main person running the study, who is often a medical doctor. They usually also have a large research team that may include doctors, nurses, social workers, and other healthcare professionals. The studies can, and often are, funded by pharmaceutical companies, as well as academic medical centers and other organizations, and federal agencies such as the National Institute of Health, the U.S. Department of Defense, and the U.S. Department of Veteran Affairs. Doctors, other healthcare providers, and other individuals can also sponsor clinical research if they have the funds for them, as studies estimate that it now costs somewhere between $161 million to $2 billion to bring a drug to market. So why go through all this trouble? Well, studies are done for many reasons. For example, to evaluate the effectiveness of drugs, medical devices, types of surgery, or therapy for treating a disease, symptom, or condition. If we never test it, how would we know if it actually works, or if it causes more harm than good? Another reason might be to find ways to prevent the development of a disease or condition in the first place, or to figure out a new way of identifying or diagnosing a particular disease, maybe even catching it early enough before it gets to a stage in which medicine can no longer help. If you're curious about what studies are being done right now, right this minute, you're in luck. Any ongoing clinical trials are, or at least should be, but we'll get to that a little later, published on www.clinicaltrials.gov, and anyone, even you, can go on there, search for a specific compound or illness, and see what research is being done about it. To make sure everything is as safe as possible, there's a lot of paperwork. Each study must be reviewed, approved, and monitored by what is called an Institutional Review Board, or IRB for short. An IRB is made up of doctors, researchers, and members of the community, and its role is to make sure that every clinical study is ethical and that rights and the well-being of the participants are protected. This includes making sure that the research risks are minimized and that the potential benefits far outweigh the potential risks for the participants. Speaking of participants, who participates in clinical trials anyway? Well, each trial must only include people who fit the particular traits needed for that study, the eligibility criteria. They may include factors such as a patient's age and gender, the type and stage of a disease, and whether the patient has had certain treatments or has other health problems, as well as other things. The eligibility criteria make sure that the people in the trial are similar and that it helps see the effects more clearly, eliminating the effect of other factors. In cases when the specific illness being studied is terminal, meaning the patients are at the very last stage of life, the eligibility criteria is often as severe as life expectancy of at least a certain amount of time, a few months or years at least, so that at least they can be expected to survive long enough for their data to be collected and measured. Clinical trials are also sometimes done in children, though, as you may imagine, even with more strict oversight. So, a natural question comes up is, why do people do this? Why do they participate? Well, some, I believe, like the idea that they're contributing to further research, especially a consideration at the end of one's life. Others are perhaps attracted by the compensation, as it can range from a couple $100s to a couple $1,000s. I just did a quick search, and I found that some offer compensation of up to $1,500, including NASA, which apparently pays $5,000 to participants willing to lie in bed for 87 days straight in order to study the effects of zero-gravity space travel on the human body. The benefits come with risks, though, of course. The whole reason for testing new therapies or treatments is because we're not sure yet what will happen. Side effects can range from unpleasant to life-threatening. So, clinical trials are expensive, time-consuming, many go on for years and even decades, and are super complicated with approvals and monitoring at every turn. But most of the time, they're worth it, either to help create a new life-saving drug, help treat a chronic condition that millions of people suffer through every day of their life, or just cash in an amazingly huge paycheck through sales profit. Whichever reason drives the trial, though, it is always a gamble, as the company or organization puts all those millions of dollars and years into work into a study. But what if it fails? Not only does it look bad and wastes a lot of money for the company, it just may be the last thing the company could ever do. Just last year's list of failed clinical trials includes examples that led to the company losing more than 60% of its stock price and eliminating jobs. In some cases, the job cuts were so severe, from 40% up to 2/3 of the entire workforce of the company. In some cases, failed trials forced the company that financed them to have to sell off license rights to their successful drugs to other companies, or downsize in space, or even sell the company entirely just to make up for the massive financial loss. Another problem arises when clinical trials fail. Oftentimes, medical journals or pharmaceutical companies that sponsor research will only report positive results, leaving out non-findings or negative findings where a new drug or procedure may have proven more harmful than helpful or shown no effect whatsoever. Basically not reporting trials that showed bad results, the ones that they did not want to report. Research into this problem points to hidden or misleading studies for all sorts of conditions, including depression, Alzheimer's, type 2 diabetes, menopause, and cancer. You can see the problem here, I hope. Only reporting good results is misleading and unfair, and it also eliminates the ability of other researchers to learn from the mistakes of their peers. Ben Goldacre, British physician, academic, and author of the book Bad Science, wrote another book titled Bad Pharma that deals with a specific issue. He argues in the book that, quote, the whole edifice of medicine is broken, end quote, because the evidence on which it is based on is systematically distorted by the pharmaceutical industry. He writes that the industry finances most of the clinical trials into its own products, and the clinical trials are often conducted on small groups of unrepresentative subjects, and negative data, or data that didn't show what they wanted to show, is routinely withheld and not disclosed. And apparently, independent academic papers may be planned and even ghostwritten by the pharmaceutical companies themselves or contractors without disclosing this information. He calls the situation a murderous disaster, but he also makes suggestions for how to improve it. If you're interested, it's a great read, and no, I'm not getting any money for this free advertisement here. Though, again, Mr. Goldacre, we would love to have you on the show, but as I understand from the email from your secretary, you're very, very busy. Anyways, the problems of the pharmaceutical industry is a can of worms we will open inch by inch, not all at once. It's just crazy. Even with all of that, though, even if participants and pharma companies are in it for the cash, and even if some studies are distorted or are unrepresentative, there are scientists out there right now trying to literally find a cure for cancer, Alzheimer's, or a myriad of other illnesses plaguing society today, so that one day, like polio and measles, we could make them a worry of past generations. I want to end in a fun fact. While clinical trials are very serious endeavors, there is sometimes a little humor sprinkled in. The researchers conducting the trial will sometimes find creative acronyms to use when referring to their work. The following is a real list of real clinical trials, their acronyms and full titles. EPIC, European Prospective Investigation into Cancer and Nutrition. Kool-Aid, Cooling for Acute Ischemic Brain Damage. CSER, Canada, Australia, Europe, South Africa AIDS Study. Christmas, Carvedilol Hibernating Reversible Ischemia Trial. Sounds like my Christmas. There's also Regional Angiogenesis with Vascular Endothelial Growth Factor in Peripheral Artery Disease, acronymized to RAVE, and a couple of others which I will not murder by mispronouncing: Torpedo, Batman, Apricot, Capricorn, and Magic. Also, Greek gods are a popular choice, with studies named Athena, Apollo, Hermes, Zeus, and so on. There is an Adam and Eve, as well as a study titled Devil. Though most of these are proper acronyms, in which, as acronyms should, each first letter of each word is chosen, but you may have noticed some did a little magic with their abbreviations, such as a study called Victory, which stands for Elective and Acute Stenting of Coronary Arteries on Express Coronary Stent System, and a personal favorite of mine, a study titled Awesome, Angina with Extremely Serious Operative Mortality Evaluation, which is definitely not awesome. The ones that I didn't say the full name you can find on the discussion post on thehealthfilespodcast.com. And check them out. They are, if not hilarious, then at least very, very confusing, yet interesting. So, from forced nutritional choices in biblical times, to forcing oranges, seawater, and sulfuric acid down the throat of sailors, to injecting mold juice extract into patients, to naming studies epic and awesome, at least we know that the future of our medicine, our health, and the health of our children is in the hands of an extremely expensive, extremely complicated process of trial and error, conflicts of interest, hidden secrets, long, complicated reports, but also people who are literally looking for how to cure or prevent cancer, and they're never ever giving up hope. So that's it for this week. If you like what you hear, subscribe, tell all of your friends, and tune in every week for a new episode. We're actually going to try to go weekly from now on. And I apologize for missing the last 2 weeks. We're buying a new house, and it's been kind of crazy. But from now on, it will be a weekly podcast. Yay! I would also love to hear your feedback about today's episode, or anything else really related to health, communications, media, research, or science. You can find me on Twitter at HealthFilesPod. The Facebook page is called The Health Files Podcast, and you can use the Contact Us page on the website, thehealthfilespodcast.com, or leave feedback in whatever podcasting app you're using to listen to the show now. It'll be really helpful to get the word out. The intro music is by the band Broke for Free. The link to their music, the website, and our various social media profiles are in the show notes. The links to all the articles I used for the show today are in the discussion post on thehealthfilespodcast.com. Thank you, and until next week.